Welcome to MOOC NPTEL course on bioengineering, an interface with biology and medicine. Before I come to some of the interesting examples of how genetics and genetic testing has made huge contributions, let me first kind of take you from the last lecture where we started discussing about genetics and genetics rules, and we were talking about law of independent assortment, where we discussed that how in dihybrid crosses, even when you are studying two different properties, those properties are getting segregated independently. And I had asked you to make some Punnett square, and I'm sure you are all trying to make those 16 boxes and trying to show that what could be the gametes will look like, but you must have realized that while it can be done, but it is not something which is very easy to do, just because you have to make too many gametes and you have to write a lot of details for them. So all of you are having mathematics and engineering background. Why not use probability rules to find out what is the probability of each type of those gametes are? And genetics in many ways is like mathematics. It's just based on rationale. If you just simply forget about is that red versus white or purple versus white, if you just forget those color names or just don't think that is what object we are talking, so you are talking about two properties. You are deriving the gene and alleles for those properties, and then you are using some mathematical rules to see that how they can transmit from one to next generation. So a good way of thinking about dihybrid crosses and multiple properties is to use probability rules. So that's what I'll talk first. So let's just first think about rules of probability, and I'm sure for this audience I don't have to teach too much what probability is. You are measuring the things in the scale of either 0 or 1. And just imagine that from egg and sperm, you are deriving the gametes. And those gametes, let's say if you have RR or capital Y, small y, those are segregating. So then they're splitting into 50%, half and half. Now from those gametes, if you see this case here, you have half capital R and half a small r from the sperm and from the eggs. And now you are deriving those gametes, so you have 1/4 probability of capital R, 1/4 probability of small r, and half the probability of heterozygotes, which is capital R and small r. So if you just think about all the crosses in this kind of probability basis, then I think many of the genetics problems which will be asked to you will become much simpler. Of course, you can still cross-check something with Punnett square, but you'll find probability rules much simpler way of deriving those. So in the law of independent assortment, we talked about that each of the gene for contrasting pair, those alleles segregate independently in the next generation. And then we have discussed that these are the dihybrids or even multihybrids, various characteristics which are there. They are passing from one to next generation, but they are actually following their own independent route. We have discussed this example of capital Y, small y, and capital R, small r. This particular heterozygote having two different type of monohybrid crosses when you are talking about two properties, the seed shape as well as the seed color. So seed color, capital Y, small y, and the capital R, small r can be your seed shape. So irrespective of these RR or YY, I'm sure you can use any nomenclature for these. So this is what the results we have seen last time. This is what I asked you to derive. And based on this particular phenotypic ratio, which is 9:3:3:1, we concluded that when Mendel was doing the experiment, he thought and hypothesized two possibilities, whether the gametes could be assorting dependent way or independent way. If it was dependent way, then probably ratio would have been 3:1. But just because in this time he found 9:3:3:1, then it was considered that they are assorting independently. And you are asked to make these chart using the Punnett square. So now let's think about the same cross, and then let's see, can we use the probability rules, and then those can be used to derive these gametes, right? So let's think about two monohybrid crosses. In all the genetics class, it will be a good idea that all of you are active. You are having your notebooks and pen in the class. Even the simple thing which we do, you are doing it, and sometime I will leave it half the way and I'll ask you to complete. So please start making some crosses. So let's take two properties, let's say seed shape and seed color. So we are doing two separate monohybrid crosses for seed shape and seed color. Very straightforward. I think we got these gametes, which is half, 50% are in the heterozygotes, and then we have 1/4 of the homozygous, dominant or recessive. So now let's think about the, how best we can use these numbers now for calculating the probabilities. So now if you multiply these individual probabilities for RR and YY, so you can start deriving these gametes, what will be their probability. The first one 1/16. All right, so what you can see now that if you would have done Punnett square, probably it was much more tedious for you to fill that Punnett square. But now if you are looking at individual probabilities first and then you are multiplying those, so now you can derive each of the gamete, what will be the genotypes and what is their probability in the F2 generation. So I think in this way, if there are different properties are given to you for a dihybrid cross, then you can first try to derive their individual crosses and then use those calculations and probability values to derive the dihybrid cross probability. Now the questions which will be asked will be something like this, that what could be probability of the F2 genotype for a gamete which is capital Y, capital Y, capital R, capital R. So to find out this one, you have to see these individual values and then you have to calculate, and then you can tell that this is probably 1/16 probability you have for this particular genotype to appear. Same for the next question, where it is 1/8 probability for capital Y, small y, and capital R, capital R. So this is very straightforward way. I'm sure now you are much more familiar and confident that the same type of things can be done using Punnett square as well as using probability rules. So now we can just simply compare the two which we have learned. This is one which we have seen in the Punnett square, and now these individual values which we have derived from the probability rules, and both of them are going to show the same ratio of law of independent assortment, 9:3:3:1. Now I think let's have some small simple questions for you. We talked about some albino child in the family. In this case, if we have both the parents are heterozygous for the albinism, what will be the probability of the child being albino? Straightforward. Let's think a little bit a different terminology now. Another disease which is achondroplasia, a type of dwarfism. In that disease, if both one of the parent is homozygous recessive and other is heterozygous, please do that. And you can use any of the nomenclature for doing these particular type of crosses. Right, correct answer, half. So you have half the child or progeny who will be dwarf, and this is actually one of the cases. Dr. Mike N, who was a scientist, and he himself was suffering from this disorder of achondroplasia. So these disorders, one would see many times in the human population, and when you investigate that, what could have been the cause, what could have been the family history, after looking at those analyses, then you may realize that some of these things are actually already part of those pedigree and those particular genetic inheritance of those families. So I'm sure now you are kind of familiar with this cross. I think I had seen one hand up a while ago for a question. I can now take a question. All right.
Multi-hybrid cross. So what's it supposed to be?
What does that mean?
Like many capital R, small r. Apart from that, is there something else?
So he is just trying to clarify that what this multi-character crosses means, and what this statement ideally, how to simplify this. So if we are looking at different properties, if you think about classical Mendelian experiment, we were talking about one property at a time, tall versus short. The flower looks like purple versus white. So we are looking at one property, one characteristic at a time. Now if you look at multi-characteristics, different properties at the same time, those could be dihybrid if you are looking at two properties, could be trihybrid, which can be three properties. Now how those particular properties are getting inherited from one to next generation. So that is what the statement meant. And then I showed you that two properties we are doing the dihybrid cross, and in which way we can split this particular cross into two monohybrids, and then we can derive the same values. So now we will solve some genetics problems. Try solving these sums using the probability rule that you have just learnt. Of course, you can do using Punnett square as well, but do not use that, because otherwise it is going to take you longer time. And you will see that how easily and quickly you will be able to solve some very complicated genetics problem using these probability rules. My TA will assist you, but as I have mentioned earlier, please solve them yourself first before checking these solutions. So let us begin with the first question. Consider a dihybrid cross occurs between capital Y small y, capital R small r heterozygotes. What will be the probability that the genotype of the offspring is capital Y small r small r? The genotype of F1 individuals in a trihybrid cross is capital A small a, capital B small b, capital C small c. Assuming independent assortment of these genes, what are the probabilities that F2 offspring will have the following genotypes? flower position, stem length, and seed shape were three characters that were studied by Mendel. Each character is controlled by independently assorting genes and has dominant and recessive expression. If a plant heterozygous for all the three characters is allowed to self-fertilize, what proportion of the offspring would you expect in terms of probability to be homozygous for the three recessive traits? Phenylketonuria or PKU is an inherited disease caused by a recessive allele. If a woman and her husband, who are both carriers, have 3 children, what is the probability of each of the following? All 3 children are of normal phenotype, one or more of the 3 children have the disease, all 3 children have the disease, at least one child is phenotypically normal. Three characters, leaf color, seed coat color and stem shape are considered in a cross between two plants, capital P small p, capital Y small y, capital I small I, cross with a small p, capital Y small y, a small I. What fraction of offspring are predicted to be homozygous recessive for at least 2 of the 3 characters? In the last few lectures, you have studied the two main Mendelian laws, law of segregation and law of independent assortment. We have discussed some very interesting examples of Mendelian genetics. You have also seen that how many human traits follow Mendelian patterns of inheritance. But you have also seen that how many traits do not follow the Mendelian pattern of inheritance. For example, in case of human blood groups, there are more than two alleles. The blood groups are not just A or B, they can also be codominant, that is A and B, or else they can be neither A nor B, that is O blood group. We have also solved many genetics problems together. You might have realized how easy it is to solve the problems using probability rules rather than drawing the Punnett squares. That is all for the day. Thank you and see you in the next class. Thank you.